Just a reminder, everybody at the desks have mics, so please keep your mics muted for the duration of the talks today, and then everybody on Zoom as well, if you could please mute your mics, especially as you're joining. Yeah, just a quick disclaimer, this event is being recorded, so if you have any questions about this, you could please contact, his contact isn't here for some reason, but Will Xiao. Just let me know at the end of the meeting if you have any questions, but usually we record our seminars and we'll post it on YouTube, so you might be famous. Our schedule for today, I guess we're in announcements, and then we'll have a 15-minute introductory trainee talk, followed by a featured talk that we're very excited for, and then afterwards we have pizza and drinks and a little social for you guys to all get to know each other and chit-chat. I'd like to thank our VanBug organizing committee, Dr. William Xiao, Dr. Amy Lee, and Dr. Faraz Hach, and then all of our lovely volunteers here. We really appreciate your support, especially the faculty members. We couldn't do it without you. And next, quick promo for VanBug. Here are our 2 upcoming events for the rest of the year. We have a talk scheduled for February 19th and March 19th, and we hope that you guys will come because I think they'll be very fun and exciting. As well, you can subscribe to our mailing list on our website. So to do that on our website, go under contact and click on the link under mailing list, and you can fill out a form, and you'll get emails about all of our upcoming events and then also anything interesting in the bioinformatics space that we think you guys might like. As well, we have a calendar that you guys can subscribe to with our events. We have Instagram, X, and YouTube, where we promote things, and again, we will upload recordings of our talks on YouTube. As well, for our next season, you guys can suggest speakers. If you have any PIs or anybody you are interested in hearing from, we can get them in Vancouver. We also welcome international speakers like our lovely speaker today, so dream big. Feel free to suggest a speaker if you have literally anybody in mind. Next, we'd like to thank our sponsors, Graduate Studies in Bioinformatics at UBC and SFU. So weird. Genome BC, SFU Big Data Hub, the Michael Smith Laboratories, Langara College, and Stem Cell Technologies, and the Vancouver Prostate Center. Next, shout out to our friends. We are affiliated with ISCB, and then our sister groups Monbug and Torbug. So for members of the Zoom audience, if you have any questions, we ask that you ask your questions in the chat, and we have volunteers monitoring the Zoom meeting, and they will voice it out for you. Okay, so now I'd like to introduce our student trainee talk for the day. It's Mehdi Esme. He's a PhD student in computer science at the Hotch Lab at UBC, and his talk is titled Deep Learning Model for Scoring Circular RNAs and Interpreting Back Splicing. So yeah, do you want to come up? Okay. Cool. Yippee.
Hello everyone, my name is Mehdi. I'm a PhD student in Dr. Haj's lab, And this was part of my master degree in bioinformatics program. The project is called Deep Learning Model for Scoring circRNAs and Interpreting Backsplicing. So as you might know, after transcription, the pre-mRNA goes through backsplicing, which you can see here. It removes the introns from the pre-mRNA. It becomes the mature RNA, but it can go through backsplicing as well, which creates these circular RNA loops, which we call circRNAs. They are very resistant to degradation, and they're found across species, and they're also often cell type or stage specific, which makes them great candidates for biomarkers or targets in different diseases such as cancer. Previous studies showed that backsplicing uses the same machinery and signaling as canonical splicing, but what's different here is that because of the proximity between splicing sites, the backsplicing is happening, and this proximity can be driven by both cis elements such as complementary sequences or trans factors such as RNA binding proteins. circRNAs have a lot of functions. I have a few examples here. They can interact with other RNAs. For example, they can bind to microRNAs and reduce their effect. They can also regulate translation or change the stability of other RNA molecules. They can bind to proteins and produce these protein complexes, and in some cases, circRNAs can also be translated into peptides or proteins. They can interact with DNA as well and influence transcription, splicing, or genome stability. And as an example of their importance, I want to show you this study about prostate cancer. They studied multiple prostate cancer, and they showed that more than 7,000 circRNAs are expressed, and more than 11% of them were essential for the cells, which is a very high number. It's around 700 circRNAs that can be targeted or studied. For identifying these molecules, there are different methods to detect them from RNA-seq data. As you can see in the figure, these methods try to find circRNAs based on these reads that are coming from backsplicing junctions. And the overall, summary of these methods is that they filter out the reads coming from linear RNAs and then remove some other reads that are coming from different events and detect backsplicing. And there are many tools doing that. And for example, CircMiner from our own lab, or FindCirc or Siri, and even STAR has an option that can detect circRNAs. But these tools usually have limitations. For example, false positive is a big problem in circRNA detection. And because of mismatched reads or PCR artifacts, structural variations, gene fusion, and other sort of false positives. And because of the low read support of these circRNAs, it's very difficult to find these false positives or even do any statistical analysis on these circRNAs. There's another option, which is treating the samples before sequencing with RNase-R, which removes most of linear RNAs, but this is very expensive and needs a lot of material, which is not possible for patient samples. So there are these models on top of the detection tools, such as circCNN and DeepCircCode, which based on the sequence coming from backsplicing sites, they classify whether this event is false positive or true. And it's just based on the sequence, so it's not based on the RNA-seq data. Existing method use mostly CNN, and they're motif-based. But what we hypothesize is that, as I showed you before, if we use both motifs coming from RNA binding protein sites and sequence similarities, we will get better accuracy and better interpretability. So we have this architecture for a model that we get the intronic sequence from acceptor and donor site, and we embed them with a convolutional layer, and we get the strongest signal with max pooling. And then after that, we have the upper branch and lower branch. The upper branch is, again, motif-based approach to get the RNA binding protein sites, and the lower branches for detecting sequence similarities and complementary sequences. And then we use a dense model later as a classifier to score and classify these events. For training this model, we used a data set generated from the existing databases. We use circ-based and circRNA-DB. We have two data sets. The first one is the approach that circ-CNN paper used. It's generated, like we get the positive samples from these databases, but we need to generate negative samples. So they generated negative samples from the transcripts that have also circRNA. But this is a little bit noisy because we have observed that whenever a transcript is generating circRNA, it's possible that like for all the exons to generate circRNA. So it's the negative set is going to be very noisy in this approach. So we came up with the second approach where we generate the negative samples from the genes that are not in these two databases. And after training, we like tested them on these data sets. So we split them into train and test. And you can see in the first data set, the scores are very similar. But the second data set, our model is performing much better than the circ-CNN. And if you look at the recall, we have like a much higher recall, which is very important because this tool is going to be used on top of detection tools. So we want to get as much as positive samples we want because we know like most of them are positives. For assessing the model on the real world data sets, we use the cell line data sets from the prostate cancer study that I showed you before. And we classified the events as low support, middle support, and medium support, and like high support. And you can see here that score for the high support group is higher than the other ones, but these results are not consistent in other cell lines, and we're trying to come with a systematic way to find the false positives using our model. For interpretation, we extracted the weights from convolutional layers, and we made this positional weight matrices, and we used motif comparison tool to extract the proteins that are interacting with these motifs. We implemented this pipeline, and I have an example here that we got the top motif from splice acceptor site, which is a AU-rich element, which is known to bind to these proteins AUF1 and TDP43, which are known proteins that make circular RNAs in different diseases. In conclusion, in this project, we developed a method to predict circRNA from only the intronic sequence. It learns both from protein binding motifs and the sequence similarities, and this framework provides a scalable and interpretable way to study RNA, circular RNAs. And at the end, I want to thank my supervisor, Dr. Hatch, our collaborators, and my lab mates. Any question? Yeah.
You mentioned about the type of cell line. Are there any similarities between the cell lines?
Oh, okay.
Yeah. You had mentioned that there was you found that you weren't able to get great results with specific types of cell lines. Is there like a common thread between those cell lines? Are they from like a certain type of cancer or a certain type of tissue?
They all from prostate cancer. They 4 prostate cancer cell line. But the point is like we're just looking at the let's say correlation between the score and the number of reads support here. But what we observe later is that even there are some events that they have many reads supporting that there's a back splicing, but they're not actually back splicing. They're coming from different types of events, for example, structural variations, or even like they're mismapped, or they're coming from like repeated regions in the genome. So they're just like the number of reads support doesn't mean that it's a true circRNA. So like it's actually like we want to find a way to just show that this is like systematically show that these are not true. Any other question? Thank you.
Why is it still on?
Thank you, Mahadi, for that talk. That was really interesting stuff. And yeah, so now I'd like to introduce our featured presenter, Dr. Iman Hajirasouliha. Thank you so much. So his talk today is titled A Modular Metagenomics Analysis Pipeline and Microbiome-in-a-Bottle. I'm very excited for this talk, so thanks for coming. A quick bio, Dr. Iman Hajirasouliha is an associate professor of systems and computational biomedicine at Cornell Medicine. Him and his lab team are passionate about developing new algorithms, machine learning and deep learning methods, and their applications in genomics, metagenomics, and cancer research. Some of the current projects in the lab include characterizing human genomes and metagenomes using multiple sequencing platforms, quantifying cancer evolution, and the study of tumor heterogeneity using genomics and imaging, and AI in vitro fertilization. So yeah, thank you so much for coming, and again, very excited to hear this talk.
What? Yeah, you can see it. Right? It's good. Okay. Yeah. Okay. Thanks for the introduction, and thanks for inviting me, especially Faraz. He and I shared the same lab at SFU a long time ago, so we were classmates. It's good to be back in Vancouver. So today I'm going to talk about modular metagenomics analysis pipeline, and also a new project in the lab, Microbiome-in-a-Bottle. So essentially the presentation has 2 different parts. So I just want to go quickly and also introduce some other projects in the lab, because my lab does a lot of different things. It's a very diverse lab. We do a lot of genomics, like computational genomics, metagenomics. So we've been developing algorithms for characterizing structural variations, assembly of structural variations, and some metagenomics applications that I'm going to discuss today. We also do basically AI in medicine, and that's another focus of the lab, mostly in women's health. So we work closely with our IVF clinic on different projects in fertility, ovarian diseases, mainly using AI to optimize the outcome. We also work a lot with pathologists on using AI for digital pathology. And also recently we use AI for genomics too. So a lot of genomics, but also a lot of AI in medicine in the lab. Today I'm going to mostly focus on metagenomics and especially the bioinformatics aspect of some of the projects we are doing, because it's like a Van Bioinformatics group. And it's also like a unique talk that I'm giving, because normally I give presentations of published work mostly, but today I'm also talking about some of their unpublished work and some sort of half-baked results. So I know there's a discussion later. We can brainstorm and discuss about these projects. But also I want to advertise tomorrow I'm giving a talk at SFU. The talk tomorrow is mostly on the AI side, still like genomics, but also I'm going to touch on some of the applications of AI in women's health and fertility. So with this introduction, I'm going to introduce my talk. I begin with microbial communities. Why do we need? Because now we have metagenomics, we have tools to sequence a lot of genomes at the same time, look at the communities of microbes. Why do we study them? And okay, one thing is we want to see who is in the community, what are the species that are present, and that's an important problem in general. But also we are interested to see what are they doing, because there are a lot of health impact because of microbes. There are, different diseases that are linked to different diseases, but also, for monitoring outbreaks like COVID recently, or, in biotech industry, a lot of applications. Basically, we want to know these communities and we want to characterize them. And it's only knowing what is there, only looking at, like some small markers, I think is not enough. So we want to do like a lot of sequencing. We want to see exactly what is there, what are the basically implications. So that's why metagenomics has become very important. And in the bioinformatics community, a lot of tools, are being developed to do metagenome analysis. And often these communities are very complex. There are, a lot of species together. It's like a mixture, so it's some of them are known, some of them are unknown. So it's really hard to analyze these communities because a lot of low abundance species might be present. It's a very complex mixture. But in a way, we have now ability to generate a lot of data. As you probably all know, we have sequencing platforms. We generate a lot of data. Now we need reliable and robust workflows, essentially bioinformatics tools that can, identify these species, identify, the functional roles of the species and, the communities. So we want to essentially analyze data with reliable, robust tools. So people in the community, like in bioinformatics community, have been developing a lot of tools, right? So this is like a busy slide, but you can see there are 19 tools for different applications in metagenomics, from taxonomy profiling, assembly, a lot of, pipelines, to do metagenomics. But as you can see and imagine, not all of them become popular, but it's still, for good reasons, people still develop these models and these tools because they see something is missing, right? They do benchmarking with previous tools, so it's still in a very active environment, like a research development environment, because we want to have the best tool. And, it's always, we're still not there for many of the applications in metagenomics. And another thing is, Okay, let's say like we do the sequencing, we start from raw sequencing. If you want to get to the outcome of metagenome assembled genomes or high quality strains, there are a lot of tools that you need to use together, right? So there are, you start with sequencing, but you may need to do the quality check, you may need to do some assembly. There might be like different sequencing platforms. You do taxonomy profiling, you want to check the quality of the mag, you want to bin the contigs, check the quality of the mags. So there are many tools for each of these tasks. And obviously each of these tools, they have their own dependencies. They have the problem that we see when we do like software development, right? As a result, many of them would be phased out, in a matter of years or even months. And, for good reason, some labs or some scientists develop these all-in-one pipeline. So we want to make it easier for people to use the tool. So let's just have like a one-click button tool. So you can essentially pull everything in one package, just run that package, right? But it's, as I'm going to mention in the next few slides, there are a lot of issues with these all-in-one pipelines, just mostly because they don't give us a lot of flexibility. There are often, parameters are fixed. There are not a lot of version control. It's sort of hard to maintain them. If you want to, change something, the flexibility is pretty low. So this is, I'm trying to explain this one-click analysis paradigm, especially in the context of metagenomics. This is an example. We have quality control, assembly, taxonomy, quantification, functional characterization, and mag binning. So all in one pipeline, right? But then that would introduce a lot of challenges because there are several failure points. If one of them fails, the whole pipeline fails. And it's really hard to figure out what it was, how to fix it. And as I said, not a lot of flexibility, right? So we want to rethink this analysis paradigm and sort of move back to a modular approach. We have several important tasks in our metagenome analysis that we want to do. Let's do it in a modular way. So for each task, we have a module, and they are all self-contained. They have the same architecture, so it's easy to maintain, easy to sort of document all of them. But the goal is, we want to finish the analysis, but let's have separate independent modules and do this in a modular way. So this would allow, users to essentially assemble a workflow that they want. So this is not just like a not flexible one all-in-one pipeline, but you can essentially choose which module you want for your analysis. And in fact, different analysis may require, different modules. So you don't need all of them for a particular analysis. Maybe you only need, maybe you only want to do assembly. So you can just, get the read, do the quality control on the read, and then do the assembly. You don't want to do the mag binning. So it gives you a lot of flexibility on what to choose and which ones to use. So you also have all of these built-in soft pauses. So you can decide, if you want to move to the next module. You can, in the meantime, you can explore the downstream results. Also, this gives you flexibility to implement many different tools for each module, and you can also add or implement new ones. So you can use it for benchmarking, and it gives you flexibility to expand this easily if you want to, develop more modules or add more to this. So with all this introduction, I want to introduce CAMP, which is a modular pipeline we developed. This is mainly work of my now former PhD student. She just defended a couple of weeks ago, Lauren Mack. And there's also a manuscript on bioRxiv, which is just accepted in NAR Genomics. So it's going to come out very soon. And the whole pipeline with all the documentation is also available on GitHub, meta-camp. So the idea is that we want to implement all these modules for metagenomics in a consistent, flexible way with the same architecture. And we also want to, we also showed some use case in a pilot project. Basically, we analyzed some sequencing data from the MetaSub project. I'm going to discuss a little bit. So this figure shows all the modules that are available now in CAMP. If you go, I guess, from left to right, so the ones that are colored with lighter red, these are more like modules and tasks that you do in the beginning of your pipeline. Once you go to the right, to the more like darker colors, those are the tools that you would probably use at the end of your pipeline. And like some of these tools may use external databases. You can see that cylinder symbol on there. And the CAMP pipeline by itself also has some visualization capabilities. So you can essentially generate figures in a consistent way, graphs, when you use each of these modules. So you can see, we have implemented short read, long read, quality control, assembly, hybrid assembly, mag binning, taxonomy profiling, and it's also, as I said, it's flexible. So if you want to add more modules to it, it's very easy. You can integrate to the current environment. Yeah, so this is one example that you can see. Basically, what I said, like these, some of them are at the beginning of the pipeline, some of them in the middle, some of them at the end. And with CAMP, we can now address some of the challenges that we had in metagenomics. Metagenomics data is complex, but this metagenomics data is complex, but now we have implemented all these modules that can be used for different analysis. It's very hard to resolve species-level genomes. We still don't have tools or technology to do it perfectly. But at least with CAMP, we can get close to it because, it's always using state-of-the-art methods for each technology. And you can, implement new tools in a more flexible way. We can also do a manual curation of our pipeline. So you can, choose which ones to use, build a workflow around it. And it's very easy to do the setup. So, you can only we have like 5 command lines for each of them, and it's consistent across all the modules. So you can easily set up and apply it to your data. So this is basically one example, how we can easily set up the environment and run each tool. And it's the same for every module. So very, very consistent. So this is one example I want to highlight here. For shorted quality control, you need to filter low quality sequences, remove host reads, do error correction on the sequences. But again, all of them can be done very easily with this modular system. So this table, you can also see it in the paper. We highlight the features in CAMP compared to other bioinformatics, other metagenomics pipeline and workflows. So you can see, we are the one who is doing the modular design. The others, they don't have the modular design. It's also very easy to install. We have a very good documentation, so you can check the GitHub. It's very extensive. We have this data visualization tool. Again, consistently you can see figures, the output of your analysis. We implemented different mag binning tools. 6 state-of-the-art mag binning tools. Was also one DAS tool, which is aggregating other tools. We also do shortly taxonomic classification and virus phage detection. So we're going over to a couple of quick examples. Let's say one user has a short-read data set and they want to do some initial analysis with short-read taxonomic classification. They can just pick, two modules, right? One to do the short-read preprocessing module for quality control, and then one for taxonomy assignment. If a user is developing a new tool, again, they can write down the tool, but it's easy to implement it in this modular system, and they can essentially use the system to do benchmarking against the existing tools. If they want to do mag binning, again, you can download a few related modules and just do the mag binning. So Metasub was a project my lab contributed. This is led by Chris Mason at my institution. So this has started almost a decade ago now. But the goal is to characterize and profile microbes across different transit systems. So there are subway stations in New York City, but now they expanded to, dozens of cities across the globe. So it's a very ambitious project. They generated a lot of data. It's now like tens of thousands of samples. So this sort of motivated this project because we didn't really have tools to analyze all this data. So we want to do it in a robust way. So together with developing this project and generating all this sequencing data, we also developed CAMP to analyze the data. And I'm showing you some results, some pilot study on Metasub samples. So we picked 10 random samples, all from New York City. And you can see on this table, they are collected from different subway stations in New York. The data set is pretty heterogeneous in a way that some samples they don't have a lot of reads. They can be anywhere from 3 million to 46 million. So it's a big range in the number of reads. And so basically, let's focus more on sample number 3 and sample number 7, the one with the least amount of reads and then one with the highest amount of reads. And let's say we want to do some analysis using CAMP. So for example, we want to do shorted assembly. First, we want to do the quality control. And you can see the result on sample 3 and sample 7. You can see for the most part, most of the bases that are being lost are at the first step when we remove low quality reads. You can also use the CAMP modules to do de novo assembly on all the samples. As you can see, for the most part, we expect that if you have a lot of reads, you also get a lot of contigs, and the assemblies are better. And that's something that we see, except for sample number 4, because that was like it has the second most number of reads, but the number of contigs is the most, and also the number of bases. And I think the reason is maybe there are less repetitive sequencing in that sample. But in general, you expect to have a high correlation with the number of reads, and that's something that we see in CAMP analysis as well. We can retain some of these contigs, some contigs that have that pass a threshold of size, and do mag binning. As you can see, many of these contigs are short, so we don't retain it for mag binning. But based on the threshold that we use, we can maybe get to 10% of the contigs, 9% of the contigs to do the mag binning. And we can use CAMP modules to do the mag binning. So here are some results. In CAMP, we have 6 different algorithms implemented. You can see each of these different algorithms estimate the number of mags differently. So it's not consistent across all the algorithms. Because, for example, some of them use single gene copy markers. They tend to have a lower number of mags. So you can see, like in these examples, Metabat and Coconut, they have the higher number of mags on these samples. We can also check the quality of mags using the CAMP. So you can see, like many of the mags in our data are actually high quality, based on like different metrics that was implemented in CAMP. We can also do taxonomy classifications on these samples. And again, I want to emphasize that all of these charts and figures are also CAMP generated these figures. So you can see the output of different modules and different analysis. So you can see, so these are like the result of taxonomy classification with 2 different profiler. They are not the same as you expect. There are many species. So these are like different ranks you can see in this data set. For many of the species, these are unique to only one of the classifiers. And you see it for other ranks as well. So the table sort of summarize the results on this data. So some takeaway from CAMP on this pilot data set. Again, metagenomics is very complex. There are so many species. It's some of them are low abundance. It's hard to sort of analyze, and as a result, almost no agreement between species for the different tools. So it's and CAMP sort of shows this with visualization and all of that. So why CAMP? What CAMP doesn't do now? I guess one thing that is missing which is important, is this multi-sample co-assembly and co-binning we are working on. And there are also some other applications that are missing. So we have some collaborators that are working with us to implement those as well. Another thing that we are excited and we started looking at is to build an AI assistant for CAMP, meaning a user would select the module that they want to use for a particular analysis, or they want to learn about CAMP, what CAMP does. Can we have an AI that can sort of help the user pick the modules, assemble the workflows, kind of what CAMP does, but with the assist of an AI. And this is a project, an active project in the lab. We call it Bootcamp, which is an AI assistant for CAMP users. This is led by my postdoc, Wei Wei. And the idea is to basically help users use CAMP. So CAMP was designed to help metagenome analysis. But now, especially for users that are not familiar with metagenomics, they don't know exactly, if they have long reads, what kind of method they need to use. The AI would tell us or guide us what kind of module in CAMP they can pick and how they can assemble the pipeline. This is still going on. It's still like an ongoing project. I don't have a lot of result to show. I just want to sort of show some examples of what the AI assistant can provide. For example, if you ask, what is CAMP? Because it has studied, all the documentation, GitHub in CAMP gives you the answer. But if you want to say, how can I do a long read assembly? Gives you exactly what kind of modules you need to use, how to set up the pipeline, and do your long read assembly. Okay, so to summarize, CAMP is a modular ecosystem we developed in the lab. We are excited to work with researchers who want to use it or expand it. And CAMP is currently available on GitHub. And we are also building this AI-assisted version Bootcamp to sort of enhance this. Okay, so this is the first part of my talk. The second part is another project we started a couple years ago, still unpublished. We call it Microbiome-in-a-Bottle project. So going back to my intro slide, we want to study these microbial communities because they are important. But we don't have a lot of high-quality genomes or reference genomes, right? To do benchmarking, to sort of assess the quality of our results. So that's a problem. So typically when we do the study of complex microbial communities, you start with some sample, you extract DNA, you do the sequencing, and you do downstream analysis to, for example, assemble the genomes, right? But a main challenge in this field is that there is no universal reference genome, okay? In fact, like many of the species are even not known, so you don't know what is in your community. The communities are diverse and uneven in abundance. For the most part, like when you have a sample, there might be, like a lot of some high abundant species in them, but then there might be like a very long tail of low abundant species. And also, like within a species, a lot of variations for like strain level variations. The taxonomy profiling is inconsistent. I showed some example in the MetaSub project. So it's not consistent when you use different profiler, different databases. And it's hard to do genome assembly in metagenomics because, there are typical problems of repeats, closely related strains, right? So in genomics community, like there was a project, Genome-in-a-Bottle. I was actually a part of it when I was a postdoc some time ago. And the idea was to use multi-platform sequencing to generate high quality reference genomes, okay? This project is still going on. So there was a main paper. Basically, they generate a lot of sequences with like short reads, linked reads, long reads. And they used all of these sequencing to essentially build a high quality reference genome and also like benchmark data sets for variant calling, structure variation, and so on. So we wanted to do the same thing, but with real metagenomic samples. We want to do a lot of sequencing on different platforms and essentially use all this data to build a strain level reference genomes for metagenomics. So just to sort of remind the audience here, we have technology now that can do like sequencing very, very quickly, pretty efficient, like inexpensive relatively. For example, we have short read sequencing. This is very popular. You can do Illumina short reads very quickly, high throughput fashion. But the reads are short, maybe 50 base pair to 200 base pair. But you can also do long read sequencing now. It used to be more expensive. Now the cost is down, so it's more affordable. It's more popular, especially PacBio, Oxford Nanopore. You can generate 5 kb to hundreds of kb long reads. There are also some synthetic long read technologies, not very popular compared to actual long reads, and there are some additional challenges. But those are also like pretty inexpensive. They don't give you actual long reads, but you can essentially synthetically build long reads from short reads that are barcoded and originated from the same long molecules. So with all of these technologies, they have like different pros and cons. Some of them have higher error rates. Some of them may have lower error rate. The read lengths, the chemistry. So there are differences among different technologies. Similar to what Genome-in-a-Bottle did, we want to leverage the diversity in sequencing platform and technology to build high quality metagenome references. And we can use this, the result of this project to guide, benchmarking metagenome tools, essentially it's like a service to the community because, they can use our data to benchmark, the different methods. So currently the options that are available are mostly doing simulation, which, as you know, cannot capture, the complexity of metagenome data. They are very limited. You can generate like high quality, genomes, but it doesn't capture the complexity. There are some mock communities, especially Zymo Research provides some, but usually they only have a few species in them. And again, they are not very complex. There are also some consortia scale project that generated a lot of data on real samples, but there's no ground truth. So it's hard to utilize them if you want to do benchmarking or if you want to do comparisons. So we wanted to start with some high quality real data, do a lot of sequencing across different platform, build like mags, high quality mags, strain level species, and provide it to the community to for like additional downstream analysis or for benchmarking. So this is the objective of this project. We want to do 4 gold standard benchmarking data sets for metagenomics. We use a real human gut microbiome, the collaboration with Mason Lab and also Zymo Research. So they did some help with the wet lab and data generation. So we use 2 DNA extraction protocols, 2 replicas. We use 5 ultra deep sequencing technologies, and we did a lot of sequencing. So it's like a very deep sequencing for all of them. Especially we did Illumina, we did Illumina HiSeq, we did Oxford Nanopore long read. We did sort of like a linked read synthetic long read technology, universal sequencing technologies, TELSIC, which gives you barcoded short read that you can generate synthetic long reads. We also did loop genomics, another synthetic long read technology. We also did phase genomics HiC. So as you can see, it's a diverse set of technologies. The only thing that is missing here, and then we want to do it in our sort of second phase of the project, is PacBio Hi-Fi, because gives you high quality long read as well. And I'm going to discuss some of the results of ONT, which I guess like most of the mags were not high quality just because their quality of reads were not that great. So we generated a lot of data, and most of the data is already available on data repositories like SRA. But we will essentially release all the data very soon. But for the most part, it's available now. So in practice, the goal is to, as I mentioned, we collected these samples with the help of Zymo Research. We want to leverage all these sequencing platforms to do high quality assembly of mags and release the mags. And throughout this process, so this is like an ongoing project. I don't have the final results. I'm showing you some of the results here. But throughout the process, we also wanted to compare, different sequencing platforms. If we are using the same sample on 2 different technology, for example, Illumina short read versus long reads, would the result would be the same, or there are difference for different applications. So one thing that we notice, as you can imagine, with short read assembly, and again we use CAM for all of this analysis, right? For short read assembly, we could recover some high quality mags. But for the most part, they remained fragmented, and we couldn't really resolve the strains. Okay, so I'm sort of like highlighting some of the results on one of the data sets. When we utilize link reads, the TELSIC data, we could actually reconstruct even higher quality mags, which is promising with these technologies. So we get higher number of mags and also higher quality mags. And, like, again, like even with short read and link reads, essentially they're all short reads, the agreement were not perfect. Only 68% of discovered species were seen in both technologies. This is something that we are investigating now. Even, for the most part, for high abundance species, they overlapped a lot, but it's still, like sometimes, in different replicas, even that was not the case. Yeah, so this was one thing that I wanted to highlight, that not all these technologies, not always they agree with each other. With long reads, we could generate, mags, and they were, like more contiguous in a way, like they are long. We get longer contigs, but many of them were not high quality when we used Oxford nanopore data, and many of them had contamination. So this is you can see, like the overall score of mags from Oxford nanopore from long read data was lower compared to short read and link read. And the contamination was higher, but as you can see in the lower bars, they are more contiguous because we had long reads, and they could resolve many of the repetitive elements in our data. So I guess this sort of shows each, and this was something that we would expect, right? That each technology has some strengths and some weaknesses. Can we combine all the technologies, build some strategies to, build high quality mags, more high quality mags when you combine all the data? So one strategy that we used for our data was to use short reads and link reads to build an initial data set of 71 high quality species. Then we went back, we looked at the existing reference genomes for these species, and then we mapped the long reads. So we started with building mags from short read and link reads. Then we mapped all the reads, long reads that matched those references. And then we supplemented, we added reads from short read and link reads, added to the coverage, and do the assembly again. So this is sort of like a strategy that we use, short read, link read mags first, and then added the lower quality but longer reads. So I'm going to highlight, a few more results. Again, this is like a work in progress. We are sort of exploring other strategies as well. But again, just want to highlight, short read, link read, and long read species, even on estimation of abundance of high abundance species, they are not always the same. So the orders are different, the level of abundance are different for each technology. Even, this is like the same data. And also something that is interesting is that a large fraction of the reads, a large fraction of long reads, cannot be assigned to any of these 74 species that we built. So how can we combine data from multiple sequencing technologies? I mentioned there are a couple different strategies. One thing that we used was one thing that you can do is to do the assembly all with long read, for example, and then do some polishing with short reads, or we can, we can use short reads and then supplement it with long reads. We were actually pretty successful in sort of building some of these high quality references or some of these strain level genomes when we combine the technologies. So I'm going to highlight one example here. You can see it's exactly like a circularized DNA. So we could assemble it into one single config with our strategy. But we are sort of trying to build more high quality contigs now. So we already have a good set of outputs of contigs, and many of them are have only like a few contigs. So just to summarize the Microbiome-in-a-Bottle project takeaway, doing this hybrid sequencing, similar to what we did in Genome-in-a-Bottle, is very helpful. With the introduction of CAMP, we use CAMP for all of this analysis, and I think that's a good way of organizing a software project using metagenomics, because gives you a lot of flexibility, and this modular, and also like the visualization aspect was very helpful for us, because we could always see what's going on, and try different strategies, try different workflows, and all of that. So hopefully, we are writing a big manuscript describing all the results. Hopefully it will be out soon. And the data is, for the most part, available. Okay, I think I'm going to stop here. I want to thank all of you again, thank the organizers, and thank all the collaborators on this project. Especially shout out to Lauren, my student, who was leading all this effort pretty much all by herself. It was very impressive PhD work. Thanks again.
I have a question about CAMP. So with workflow systems, they have their advantages and disadvantages.
Sorry, can you speak louder?
Oh, does this not work? Can you hear me? Sorry. Okay. So with different workflow management systems, there's advantages and disadvantages, right? For CAMP, what considerations went into choosing SnakeMake over Nextflow? There's been a push for more Nextflow development here.
Yeah,
so
yeah.
I'm curious. Yeah.
Well, that's a good question. I get asked this a lot. We can do Nextflow too. It's not a... I think in the next version, we may do Nextflow as well. Yeah.
So right now, you're also leaning to adapting Nextflow as the choice of tool then?
Yes.
Okay. That's all.
Hi. In a sort of similar line of questioning, I guess, what do you use to manage environments for CAMP? Do you use Docker or Singularity?
So we don't have it yet, and that's a missing part of CAMP. Yeah.
Is it just Condo REST?
Yeah, it's Condo now.
Oh, okay.
Yeah.
Okay.
Hi, I have a quick question about AI assistant that you mentioned. I was thinking that in theory, if documentation was good enough, that in no time agentic systems would be powerful enough to use software like CAMP. Do you think there are specific advantages of like a so specialized genetic assistant?
Yeah, so that's something that we are actually exploring with this new AI system. Yeah. I don't have the definite answer for this yet.
Thank you.
Yeah, yeah. Thank you for the talk. You mentioned that a lot of long reads didn't map to those species. Is it because of the contamination that you mentioned, or is it that like the tools are not developed for long read sequencing?
I think it can be a combination of both. I think mostly because this data specifically were not very high quality long reads. Yeah.
Thank you.
Hello, thank you for the talk. I have a couple of questions. For the project Microbiome-in-a-Bottle, why the human gut microbiome? Like, was there a specific reason to choose that, or?
It was easier for us to obtain it through our collaboration. So initially we wanted to do a few more, but we may do, like soil or, other microbiome as well. But it was just, the project became very big because, there was like a lot of sequencing data, a lot of tools. Yeah, that was the main reason, I think.
Okay, and also for camp, for the boot camp section, like talking about why the human gut, like for example, if you have a specific microbiome environment, like could you ask which type of modules would be better to use in camp for this type of organism or this type of strains that I know that may exist in my metagenomic analysis?
Yes.
Okay.
Yeah, you can.
Oh, it's me. So I'll spice it up a bit. So we have a question from Zoom. I'll save my questions. So the question is coming from Will. He's asking, what's the source or the reason that you have higher levels of contamination in your long reads sequencing data?
I don't know, actually. Yeah.
I'll ask one from my own questions. So you mentioned that you use short and link reads before actually using the long reads, right? So you build assembly. So why not just error correct your long reads, start with that, and then use your link reads for extra length and use your short reads for correction of the long reads?
So there, as I mentioned, there can be like several different strategies, but this was something that I guess we got the best results when we tried it.
Me? All right. Yeah, so thank you for the talk, and I'm sure many labs, many people here are taking note of your bootcamp idea, and they will, I'm sure, run with it. But my question is about the microbiome work. So, as you know, the SMD problem is near and dear to my heart. What would it take to have a microbiome sampled, sequenced, and assembled? So where is the weakest link at the moment? Is it the sampling? Is it the sequencing technology? Is it the assembly algorithms? Where should the field concentrate to solve that problem?
I think all of them. As you know, it's improving a lot, with sequencing, right? But it's still, we are not there yet. So I think a combination of all of that. So I personally like this approach of hybrid assembly of using, multiple sequencing platforms. It's expensive, right? And it's I mean, it's a challenge because, you have to do a lot of different library prep. It's time consuming, it's expensive. But I think if you have like a diverse set of data points and, different technologies, you can do it much better. That's my experience, at least with this data. And we see this in human genomics too, right?
Sorry. You mentioned that you use short reads plus linked reads for the microbiome. How much benefit does the short reads give us on top of the linked reads? Linked reads are also short reads, right? So what do we get out of it from? Thank you.
Yeah, so I think it gives us a lot more coverage. So we could basically recover a lot more, I guess not a lot more, but it's still, like several low abundance species with short reads. And it would help, like build this consensus. So the coverage was very high for short reads.
Hi. I'm curious how you measure success in your Microbiome-in-a-Bottle projects. Is there like a particular number of reference genomes that you aim to collect per year or something like that?
So the number, the quality of contigs, and essentially if we can get to circularized DNA.
Just curious, how many like reference genomes do you have today?
We have about 7.
Hello, thank you for the talk. I'm here. Hello. I was gonna ask about the software engineering part of the first part of the talk. It all boils down to, I guess, reproducibility with these kind of projects. And modularity and reproducibility kind of does conflict with each other if you do too modular and if you try to modularize too much of your environments, of your processes. In software engineering, there are a lot of techniques that you can use, like different architectural styles in the code. It can be different design patterns as well. But when it comes down to assembling tools that are already written, what modularity is the best, and how do you go with deciding on which modularity you should be using for your pipeline?
Yeah, that's a great question. I mean, we did the modularity based on the tasks in a pipeline. Like one module is one task, basically. So we didn't want to, like further break it down or, so that was our thing.
Can I follow up? I'm sorry.
Yeah.
Sorry. What I was going to ask as a follow-up was, there are tasks in hand, but how do you even define a task, right? You can define a task with more granularity versus I guess my question was more about that. How can you define
Yeah, so when I said task, I mean, like a specific task in a metagenomics analysis pipeline. For example, taxonomy profiling or short-read quality control. That are like very well defined. Yeah, I mean, you can make it more, I guess, granular. Yeah.
So we have Rachel next, and one more question because the pizza will get cold. Cold pizza.
Okay, I'll be super quick. Can we go back to talking about how the taxonomic classification is, I think, predicted by the tool? Like the different, like I think there was a slide on that. The different tools classify the metagenomes like taxonomically completely differently.
Mm-hmm.
So are either of these like right, or do we not know? Are neither of the tools useful?
Yeah, that's that's a great question. I think for the most part they are right, but not always, right? So there are definitely differences. So it's the same data. So there should be, I mean, ideally it should be the same, right?
So is there like a ground truth or?
No, we don't have, that's the thing. We don't have the ground truth. And that's one reason we want to do this Microbiome-in-a-Bottle project. So to just build more ground truths.
So do you think sorry, do you have an answer for me? Okay. Oh, no? Okay. So would you say the classifications are like similar enough that you kind of know what's going on, or is it just like you have no idea?
So they are not, I mean, you can see this in this figure, right? So there are definitely similarities, but still there are classifications that are unique to only one of these tools. So we don't know like which one is correct, right?
Okay, thank you.
Final. So we have ENCODE and gene code in human. So do we have anything like that in the metagenomics?
I don't know actually. I don't think so.
So for example, if I want to go and like look up for a gene name, I can go and look at ensemble ID, or like different databases are like curating them according to some standardized like pipelines. Do we have some kind of a gold standard standardization methodology that everybody uses in this field?
I don't think so, no.
Okay.
I mean, there are a lot of studies you can check, but not something standard.
Okay, thank you.
Thanks again for the great talk and engaging discussion. So this is going to conclude our talk today. There's a pizza waiting for you outside, and there are also drinks. Enjoy yourself, and our speaker will be here for another 20 minutes, so you can actually socialize if you would like. We'll see you outside.